Imagine a microscopic battle unfolding right now all around us, actually inside us.
Hmm.
A tiny invader, right?
Right.
An enemy that hides its tracks deep within our very cells, merging with our very blueprint.
Yeah.
And then a precise, almost surgical strike that targets that hidden menace.
A strike born not from our own, complex immune system, but from an ancient, really ingenious defense mechanism found in the simplest of life forms.
Wow.
A mechanism that, well, for years went totally unnoticed by us, yet holds the most astonishing key to rewriting the very script of disease.
Today we're taking a deep dive into that microscopic marvel, a truly revolutionary technology known as CRISPR. And we're exploring how it's being wielded like a pair of molecular scissors, aiming for one of those stubborn and devastating global health challenges humanity has ever faced: HIV/AIDS. When we talk about HIV/AIDS, it's really not a historical footnote, not some distant memory. This disease remains a massive global public health burden, a
Yeah.
relentless adversary that continues to impact millions of lives.
It really does.
The numbers are truly stark, truly sobering. We're looking at approximately 38 million people infected worldwide.
38 million.
Yeah, with around 800,000 deaths every single year from HIV-related conditions. And despite decades of incredible scientific progress, despite some truly heroic efforts, there's still no effective vaccine, and crucially, no definitive cure.
That's absolutely right. I mean, the current standard of care, known as combination antiretroviral therapy or CART, it's undeniably been a medical triumph for so many people.
Oh, for sure.
It's transformed HIV from what was once, almost universally a death sentence into a manageable, albeit chronic, condition for millions. This therapy works by targeting and inhibiting different stages of the viral replication cycle, essentially putting up roadblocks to prevent the virus from escaping and multiplying uncontrollably.
And it has saved countless lives. But, and this is a really significant but, CART comes with some heavy baggage, doesn't it?
It does.
Patients have to commit to taking these drugs every single day for the rest of their lives. Imagine that commitment, that daily regimen, the constant reminder of the virus lurking within.
Yeah, it's a huge burden.
And it's not just the inconvenience. There's the potential for a wide range of significant side effects, which can range from, mild discomfort to really debilitating conditions that severely impact quality of life.
Right.
And even more critically, this life-sustaining therapy is still not universally accessible to all HIV-infected individuals around the globe.
Yeah.
Many simply cannot afford it or don't have access to the necessary healthcare infrastructure.
Precisely. And even for those who can access and consistently adhere to CART, it doesn't actually get rid of the virus. It only suppresses it.
Right.
The virus, in its cunning, finds a way to hide. The major obstacle standing in the way of a definitive cure right now is the presence of what scientists call the HIV reservoirs.
Ah, the reservoirs.
These are incredibly sneaky, long-lived, and frankly still pretty poorly understood infected cells, primarily CD4 plus T cells, where the HIV genome has integrated itself directly into the host cell's own DNA.
So it just sits there silently, like a ticking time bomb, a genetic ghost.
Exactly. It's like the virus has embedded sleeper agents deep within our own cellular infrastructure. These are latent infections.
Okay.
They're not actively producing new viruses, but the blueprint is there, hidden in plain sight. And if therapy stops, even for a short period, or if the cells get reactivated by other stimuli, these reservoirs can wake up, start producing new viruses, and reignite the infection, leading to a viral rebound.
Wow.
This is why the search for an HIV cure really hinges on developing strategies that can trigger the complete eradication of these viral reservoirs, not just suppress them indefinitely.
So what if we didn't just suppress the virus or build roadblocks, but went in and snipped it out? Get this. The key might lie in something bacteria, these incredibly simple life forms, figured out millions of years ago. A microscopic arms race that has been unfolding for eons. It sounds like the stuff of science fiction, but gene editing is showing incredibly encouraging results as an alternative method to inactivate that integrated HIV DNA.
It really is.
And leading the charge, the one getting all the buzz, is the CRISPR-Cas system. Now, this deep dive is heavily informed by a fantastic review paper, A CRISPR-Cas Cure for HIV/AIDS, published in early 2023 by Mariah Hussein, Mariano A. Molina, Ben Burkhout, and Elena Herrera Carrillo.
Yeah, great paper.
They're all from the Laboratory of Experimental Virology at Amsterdam UMC. University of Amsterdam.
Yeah.
They're really dug deep, evaluating the current landscape of CRISPR-Cas-based therapies for HIV,
Yeah.
meticulously focusing on viral biology, host restriction factors, and the immense potential of a combined approach.
What's truly fascinating here, and what gives me so much hope, is that we're talking about moving beyond simply managing a disease to potentially eradicating the very genetic material that sustains it. It's a fundamental shift in medical philosophy, right? Moving from control to cure, which, in the grand narrative of human health, is nothing short of monumental.
Okay, let's unpack this, because the foundation for this incredible gene editing tool, CRISPR, actually comes from the adaptive immune response found in prokaryotes, that's bacteria and archaea.
Right.
So are bacteria basically keeping a mugshot gallery of every viral offender that's ever tried to invade them?
Precisely. It's a biological memory system, an incredibly ingenious form of adaptive immunity. Think of it like this. Imagine a high-stakes genetic game of capture the flag.
Okay.
When a virus, specifically a bacteriophage, attacks a bacterium, the bacterium isn't defenseless. It has this incredible ability to literally capture a small piece of the viral DNA from the invader.
Whoa.
And then here's the genius part. It doesn't just destroy it. It stores that piece. It integrates it into its own genome, specifically into a section called the CRISPR array.
Whoa. So they just tuck it away for later,
Yeah.
like a genetic trophy case of vanquished foes, or maybe a library of most wanted posters?
More like a highly organized archive of past threats. This CRISPR array is a sequence of repetitive DNA separated by these unique spacer sequences, which are the bits of captured viral DNA.
Uh-huh.
So yes, it's a record. And if that same virus, or one similar to it, tries to invade again, the bacterium now has a permanent record. It immediately recognizes the threat because it has the exact genetic signature stored in its array.
Right.
This allows for a swift and targeted counterattack. It's truly a marvel of natural engineering.
So they remember. But how do they actually use that record? What's the specific molecular toolkit involved in this amazing bacterial defense system?
This is where it gets really interesting and where the molecular scissors come into play. The core players in this system are the Cas, or CRISPR-associated proteins.
Cas protein.
These are the molecular toolkit. They're the enzymes that do the actual work of seeking out and cutting DNA or RNA. But they don't just cut randomly.
Okay.
They're guided by a specific instruction manual, or what we call CRRNA, which stands for CRISPR-RNA. This is a short RNA sequence that's transcribed directly from that stored viral DNA in the CRISPR array. It's complementary to the target viral DNA, and it acts like a highly precise wanted poster or a GPS system guiding the Cas enzyme exactly where to go in the cell.
So the cRNA is the key, telling the Cas protein exactly which genetic sequence to hunt down.
Exactly. Now, the specific Cas enzymes have different specialties, almost like different tools in a kit. For instance, Cas9 and Cas12 are types of endonucleases, which means they are enzymes that cleave or cut DNA.
Cut DNA.
They make what we call double-strand DNA breaks, or DSBs. Think of it like a very precise cut right through both strands of the DNA ladder.
Okay.
But here's a crucial detail for their accuracy. For these enzymes to work successfully, they need a short 2,6 nucleotide sequence called the protospacer adjacent motif, or PAM, that flanks the target DNA.
PAM.
Yep. PAM. You can think of the PAM as a secret handshake or maybe a landing pad that Cas9 or Cas12 needs to recognize. It's the enzyme's final confirmation that it's in precisely the right spot to make the cut. Without that PAM, no cut. It's an essential fidelity check.
That's a super clever way to ensure pinpoint accuracy, making sure it doesn't just snip anywhere. What about the other Cas enzymes? I heard there's a Cas13.
Yes, Cas13 is quite different from Cas9 and Cas12. This enzyme specializes in cleaving RNA, not DNA.
RNA.
And for some Cas13 orthologs, it requires a protospacer flanking site, or PFS, which functions similarly to the PAM for DNA targeting Cas enzymes. It's important to note the distinction here. While Cas13 can efficiently degrade target RNA, essentially shredding viral instructions.
Shredding, I like that.
It does not edit the genome. This means it cannot directly touch or alter the integrated HIV DNA that's hiding in those viral reservoirs, which is our primary target for a true cure. It can inhibit active viral production, but it can't erase the blueprint.
So once Cas9 or Cas12 makes that perfect, precise snip, what happens next? Does the cell just leave its DNA broken? That sounds like chaos.
Oh, absolutely not. Our cells are incredibly sophisticated with built-in emergency repair crews. When Cas9 or Cas12 creates a double-strand DNA break, the cell's natural repair mechanisms are immediately activated, almost like a frantic scramble to fix the damage. There are two main ways the cell tries to fix these breaks. The first is called non-homologous end joining, or NHEJ.
NHEJ.
Okay, this is often considered the error-prone repair pathway.
Error-prone, huh? Sounds a bit risky for something as important as our DNA.
It can be. NHEJ kicks in when there isn't a perfect donor template available to guide the repair. It essentially tries to just stitch the broken DNA ends back together as quickly as possible.
Hmm.
Imagine you've torn a page out of a book and you're trying to tape it back together without the original page to guide you. You might get it mostly right, but you're likely to end up with some missing words or extra bits.
Yeah, I could see that.
This often leads to small insertions or deletions, what we call INDLS, or sometimes even substitutions around the cleavage site. Now, if these INDLS happen within a protein coding gene, they can dramatically shift the open reading frame, which is like scrambling the genetic instructions.
Wait, what exactly is an open reading frame? Is it like the specific instruction set the cell is trying to read?
Exactly. Think of it like a very precise sentence in a book of genetic code. DNA is read in triplets of nucleotides, like three-letter words.
Right, codons.
Exactly. If you add or delete even one of these letters in the middle of a sentence, the entire meaning of the sentence gets scrambled from that point onward, or the cell might read a stop signal where it shouldn't. So, if NHEJ introduces an INDL, it can lead to a frame shift, creating non-functional proteins or no protein at all.
Okay.
It's like fixing a broken zipper by just jamming it together. Sometimes it works, but often it's messy and makes the zipper completely unusable. For HIV, this messy repair is actually what we often want because if it scrambles the viral gene, the virus can't function.
Ah. Okay, so NHEJ is the quick, messy fix that can be quite useful if we want to break something viral. What's the elegant, precise solution then?
The elegant, precise solution is homology-directed repair, or HDR. This is the pathway we aim for when we talk about truly precise gene editing. HDR requires a donor DNA template.
Okay,
This template is a carefully designed piece of DNA that contains the desired genetic modifications, perhaps a single nucleotide change, or the insertion of an entirely new gene. It's flanked by DNA segments that are homologous or very similar to the DNA on either side of the cut.
Got it.
The cell then uses this template as a perfect guide to precisely repair the break and introduce those specific desired changes. It's like having a perfect instruction manual, the exact replacement parts, and a meticulous engineer to fix something with pinpoint accuracy.
Wow.
This isn't just a technical detail, it's the difference between a messy, unpredictable fix and the kind of surgical precision that allows us to dream of truly rewriting the human genome with pinpoint accuracy.
So if NHEJ is the blunt force trauma that can disable the virus, HDR is microsurgery, allowing us to insert specific instructions or correct errors in our own genome.
Got it. Now, you also mentioned deactivated Cas variants, like a Cas that can't cut. That sounds counterintuitive. Why would you want a pair of scissors that doesn't snip?
It does sound counterintuitive, doesn't it? But it's absolutely brilliant and a testament to clever molecular engineering. Scientists have engineered what are called deactivated Cas or DCas variants.
DCas, dead Cas.
Yeah, sometimes called dead Cas. These are modified versions of Cas9 and Cas12 enzymes where their cutting ability has been deliberately switched off. They've lost their nucleotide activity, the ability to cleave DNA, but they still retain that crucial ability to bind to specific target DNA sequences guided by the cRNA.
So they're like blunt scissors, but instead of cutting, they're just tapping on a gene's shoulder to tell it what to do. That's wild. It allows for control without destruction. Exactly. Instead of cutting, these DCas proteins are often linked or fused to regulatory domains. These are like little molecular switches that can either activate or repress gene expression.
Oh.
So when the DCas guide RNA complex binds to a specific gene, the attached regulatory domain can then turn that gene on or off, or turn its activity up or down in a sequence-specific manner.
Wow.
This allows for a more nuanced control, not just destruction, but a sophisticated orchestration of gene activity. Imagine turning up our cell's own natural antiviral defenses without ever touching the viral DNA itself. It's a huge leap in control and precision.
That truly is a huge leap. All right,
Yeah.
so that's the basic toolkit. Now, having understood the molecular mechanisms, the real question becomes, how are scientists actually taking these incredible bacterial insights and applying them directly to the human problem of HIV/AIDS? What does the CRISPR-Cas cure look like in practice against such a wily opponent?
Well, the most direct approach, and the one that holds the most promise for a true functional cure, is to use CRISPR-Cas as a direct antiviral. This means aiming to inactivate or literally remove the integrated proviral HIV DNA from the host genome. Remember those hidden reservoirs we talked about?
Yeah?
This approach goes straight for them. In vitro studies, which are laboratory experiments in test tubes or cell cultures, and preclinical trials in animal models, have already shown very positive results with this direct targeted strategy.
So, going straight for the hidden viral blueprint, the integrated DNA that forms those reservoirs. But how do you pick which parts of the HIV genome to target? The virus is pretty good at evolving and mutating, right? It's like it's constantly changing its disguise.
You've hit on a critical point. To minimize the likelihood of the virus escaping our CRISPR attack, we don't just pick any part of its genetic code. We meticulously focus on targeting viral genes that have highly conserved sequences.
Conserved sequences, meaning they don't change much.
Exactly. These are the genetic regions that are absolutely essential for the virus to survive and replicate, and therefore they don't change much, even as the virus mutates. The virus simply can't afford to alter these vital sections too much without losing its ability to infect.
Give us an example of one of these critical targets.
Certainly. The HIV Rev encoding gene is an excellent example. Its sequence is highly conserved among HIV subtypes, making it a prime target.
Okay.
Rev is crucial because it helps shuttle viral RNA out of the nucleus, allowing the virus to make its proteins. If we disrupt Rev, we essentially trap the viral RNA, preventing the virus from building new copies of itself.
Right.
Studies using CRISPR-Cas9 to target the second exon of Rev in latently infected cells have shown a remarkable 20-fold reduction in HIV gene expression and viral production.
20-fold.
Yeah, that's like cutting off the virus's supply lines and severely crippling its ability to even get started. Even the RNA targeting CRISPR-Cas9 system has been shown to inhibit HIV production when targeting Rev, specifically that same second exon, by shredding the Rev RNA.
That's a significant reduction in viral activity. What other viral genes are on the hit list for these molecular scissors?
Another absolutely key target is the HIV TAT gene. TAT is a viral protein that acts as a powerful transcriptional activator, essentially supercharging the production of viral RNA from the integrated provirus.
TAT.
That's right. If you can disrupt TAT, you severely dampen the virus's ability to express its genes. CRISPR-Cas9 targeting the first exon of TAT has led to a staggering 53-fold reduction in viral protein production, specifically the CAP24 protein, which is a major component of the viral capsid in latently infected cells.
Wow, a 53-fold reduction. That's incredible.
It truly is, and here's where the strategy gets even more sophisticated.
Yeah.
Combinatorial approaches, using 3 different cRNAs, those genetic guideposts all aimed against the TAT gene, have shown sustained inactivation of all infectious proviruses in T-cell cultures.
All of them.
All of them in those experiments. We've even seen dual CRISPR-Cas9 approaches targeting both the GAG gene, which codes for core structural proteins, and the first exon of TAT-Rev, achieve incredibly durable inactivation of HIV replication in T-cell cultures for up to 128 days.
Whoa.
This led to the extinction of all infectious proviruses in those experiments. That's a monumental step towards truly clearing those reservoirs. It's like not just hitting one part of the virus's command center, but simultaneously striking at its communication, manufacturing, and structural capabilities.
So hitting multiple targets at once, the multiplexing approach, seems to be the key to really locking it down and preventing that viral escape. But what about the core structural proteins of the virus themselves, like GAG, POL, and MVA? Are they vulnerable?
GAG and POL are indeed very attractive targets because they encode highly conserved proteins that are absolutely essential for viral structure and replication. GAG forms the viral capsid, the protective shell, and POL encodes crucial enzymes like reverse transcriptase and integrase.
Right, essential stuff.
CRISPR-Cas9 has successfully inhibited viral production and infection by targeting these sequences. However, this is precisely where the viral escape issue often comes into play. If we use a single cRNA targeting GAG or POL, the virus can quickly mutate around the cleavage site through the cell's error-prone NHEJ repair mechanism.
Messy repair again.
Exactly. These mutations can then allow the virus to escape being recognized or cut again. It's like the virus quickly changes its disguise after being spotted once, making the same wanted poster useless.
Ah, the virus's countermove, a genetic feint. So how do we outsmart that escape artist? How do we hit it in a way it can't recover from?
By getting even more strategic and relentless, a much more effective approach has been to use cRNAs targeting GAG and POL in combination with cRNAs that target the long terminal repeat regions, or LTR.
LTRs.
The LTRs are critical regulatory elements located at both ends of the integrated HIV genome. They're essential for viral gene expression and integration. Multiplex targeting of both the 5-LTR and 3-LTR elements, along with GAG and POL, using a smaller Cas9 variant called SAC-Cas9, has efficiently inactivated proviral DNA. While some studies have still observed viral escape in T-cell cultures, even with these dual approaches, the principle is clear. It's much harder for the virus to mutate and escape when it's being hit in several critical, indispensable places at once.
Right.
It's like trying to change your disguise when someone's firing paintballs at you from 3 different directions. Statistically, you're going to get hit and identified.
And what about MViv? I remember MViv being highly variable, but which makes it a tough target, right? Like trying to hit a moving target that constantly changes shape.
Precisely. N, which codes for the viral envelope glycoproteins that allow the virus to attach to and enter host cells, is highly variable among HIV genomes. This variability makes it less ideal as a single target because there's a higher probability of low strain coverage and viral escape.
Makes sense.
However, even with N, combining cRNAs targeting it with GAG has shown promise in suppressing viral replication. The more ways you hit it, the better the chances of overwhelming its ability to replicate.
It sounds like we're mapping out the virus's entire genetic weak points, from its structure to its control systems. Are there other, perhaps less obvious, targets within the virus itself, maybe its saboteurs?
Absolutely. The genes encoding the accessory proteins VIF, VPU, and VPER are also being explored. These proteins are fascinating because they're not directly structural components, but they are absolutely essential for viral replication, primarily by counteracting our own cellular restriction factors. They're like the virus's special ops team, designed to disable our internal defenses.
Okay, tell me more about these viral saboteurs. This sounds like an ingenious twist in the molecular battle.
Let's start with VIF. VIF counteracts a cellular factor called ApoBec3G.
ApoBec3G.
Yeah, it's one of our body's natural, ancient defenses. It's an enzyme that normally impedes HIV reverse transcription by hypermutating the viral DNA, essentially making it unreadable and non-functional for the virus.
Okay, so it's a good guy.
It's a very good guy for us. VIF's job, the viral saboteur, is to trigger the degradation of ApoBec3G, silencing our defense. Now here's where CRISPR comes in. Researchers have used deactivated Cas9, or dCas9, that blunt scissors we talked about, fused to an activation domain.
Right, the activator.
This dCas9 is guided to the promoter region of our endogenous ApoBec3G gene to induce its expression. By ramping up our own ApoBec3G levels, even in the presence of VIF, they observed a strong inhibition of HIV infectivity. It's like giving our own cellular defenders a superpower boost, allowing them to fight back more effectively even when the viral saboteur is active. There's also some interesting work on an endogenous E3 ubiquitin ligase called CIP, which promotes the degradation of VIF itself, further enhancing ApoBec3G activity. So yes, it's not just about destroying the virus, but essentially turning its own clever sabotage against it by empowering our existing, often overlooked cellular soldiers.
So we're not just attacking the virus directly, but also bolstering our own cellular defenses. That's a true two-pronged attack. What about VPR? What's its game?
VPR is another viral protein that interacts with multiple host restriction factors. One of them is SAMHD1, which acts as a DNT PACE.
Hold on. A DNT PACE, can you break that down for us?
Of course. Think of DNTPs as the essential raw materials, the specific DNA building blocks that the viral reverse transcriptase needs to make its DNA copy.
Got it. Building blocks.
SAMHD1 acts like a biological scavenger, a DNT PACE, scooping up and depleting these crucial building blocks. So without those DNTPs, the virus is essentially starved of its construction supplies. SAMHD1 also has RNase activity, meaning it can degrade HIV RNA.
Okay, so another good guy.
Definitely. Now here's an interesting twist that highlights the complexity. Studies that used CRISPR-Cas9 to knock out SAMHD1 actually resulted in an increase in HIV infectivity.
Wait, if SAMHD1 is a defense, why would getting rid of it make HIV stronger? That's counterintuitive.
It is. This seemingly paradoxical result highlights how crucial SAMHD1 is as a restriction factor. It means that we wouldn't want to target SAMHD1 for destruction with CRISPR. Instead, we want to understand how VPAR is trying to get around our SAMHD1.
Oh.
VPR also activates DNA damage response pathways in the host cell, inducing a premature cell cycle arrest. This arrest is thought to help the virus process excess reverse transcription products, making itself less visible to our innate immune system. While an initial theory linked VPR to an SLX4 endonuclease complex in this process, inactivating SLX4 with CRISPR-Cas9 didn't abolish the VPR-induced cell cycle arrest, so there's still more to learn about VPR as a full bag of tricks.
Fascinating. And what about VP? What's its role in this molecular cat and mouse game?
VP plays several key roles for the virus. It induces the degradation of CD4, which is HIV's main entry receptor on T cells.
Right, CD4 cells.
By clearing CD4 from the cell surface, VP makes it harder for newly produced viruses to infect the same cell again or to infect other cells in the immediate vicinity. More importantly, VP also suppresses a crucial restriction factor called tetherin.
Tetherin. Sounds sticky.
It is. Tetherin is a type 2 transmembrane protein, essentially a protein embedded in the cell membrane that sticks out like a little molecular tether. It evolved specifically to inhibit virus release from infected cells, literally tethering newly formed virions to the cell surface, preventing them from budding off and spreading.
So tetherin is like a sticky trap for new viruses.
Precisely, and VPU's job is to disable that trap. But researchers have used CRISPR-Cas9, again fused to an activation domain, to target the tetherin promoter.
Okay.
By doing so, they could significantly enhance tetherin expression. Higher tetherin levels then effectively inhibited HIV production and replication, even when VPU was present and trying to suppress it.
Nice.
So boosting our own tetherin is a very promising avenue for an indirect approach to limiting viral spread.
This whole discussion about accessory proteins raises an important question. It's not just about destroying the virus, but understanding how its own clever survival mechanisms might be leveraged. It's a delicate balance, a complex genetic chess match.
It truly is. For example, we know HIV has a mechanism called superinfection resistance, or SIR.
Sir.
This is HIV's way of preventing a second infection by a different strain of the virus in an already infected cell. It's a virus saying this cell is already occupied. HIV achieves this by downregulating the CD4 receptor, primarily using viral proteins like NEF, M, and VPU, with NEF being particularly strong in this regard.
So we're almost playing genetic chess with the virus, trying to outsmart its escape routes and even its self-preservation instincts.
In a way, yes. The review highlights that to maintain SIR in HIV-infected cells when we're attacking them with CRISPR-Cas, the expression of NEF should ideally remain intact.
Really. Keep NEF.
Well, if you target and inactivate Rev function, HIV gene expression might be restricted to just the early TAT and NEF proteins. An early study observed that Rev defective proviruses, which keep NEF intact, were still able to exhibit SIR, effectively sheltering those infected cells from wild-type HIV trying to come in. This means we're learning to understand the subtle interplay between viral proteins and host factors, sometimes even allowing some viral functions to persist if they paradoxically help prevent new, more aggressive wild-type infections from taking hold. It's about strategic thinking, not just brute force. More studies are certainly warranted on the nuances of targeting NEF.
So that's the direct attack, going after the viral DNA and RNA, and even boosting our own specific cellular countermeasures. But you mentioned indirect approaches as well, which seems like a completely different angle.
Yes, besides directly targeting viral components, we can use CRISPR-Cas to modify host genes to make our cells resistant to HIV or to boost their natural antiviral defenses. It's like teaching our cells to build their own fortresses.
Okay.
For example, using CRISPR-Cas9 to edit host genes that encode the viral entry receptors like CCR5 and CXCR4 on CD4 plus T cells can effectively make those cells resistant to HIV infection.
CCR5, that sounds familiar. Wasn't there a patient?
Exactly, the Berlin patient, Timothy Ray Brown. He received a bone marrow transplant from a donor with a natural mutation in the CCR5 gene, making his cells resistant to HIV. That was a key proof of concept. CRISPR allows us to potentially engineer that resistance deliberately.
Wow. So we could potentially recreate that effect without needing a transplant.
That's the hope. These are the specific doorways the virus uses to get inside our cells. If we can alter these doorways, the virus simply can't get in. It's like building an impenetrable wall around the cell before the virus even gets to knock.
That sounds incredibly powerful, preventing entry altogether.
Yeah.
And then there's boosting existing defenses.
Absolutely. Instead of cutting viral DNA, we can use those DCAS variants we discussed, the blunt scissors, fused to an activator,
Right.
to activate the expression of host genes that naturally fight the virus. We touched on ApoBec3G and tetherin. This indirect activation allows the cell to ramp up its own antiviral machinery, turning its natural defenses into a formidable front line. It's less about direct assault and more about empowering our cells to protect themselves.
And finally, you can also use CRISPR-Cas to insert entirely new genes, right? Like custom instructions for the cell to become a mini pharmacy.
That's one of the most exciting aspects of precise gene editing. CRISPR-Cas can be used as a precision delivery system to integrate specific genes of interest into host cells using the HDR pathway.
That precise repair method.
Exactly. A fascinating example discussed in the review is a dual adeno-associated virus, or AAV, CRISPR-Cas9 mediated approach. This integrated antibody genes into the B cell loci of mice to express anti-HIV broadly neutralizing antibodies, or BNAbs, in vivo.
Broadly neutralizing antibodies.
Yeah.
Means they can fight off many different strains of the virus, right?
Exactly. And the result of this approach was quite significant. High titers of these anti-HIV BNAbs that could neutralize infection-competent pseudoviruses in mice. Imagine the potential. Instead of patients needing regular infusions of these antibodies or taking daily pills, our own bodies could be engineered to continuously produce potent antibodies to fight HIV. It's a significant step towards long-term, self-sustaining protection, a truly one-and-done approach for passive immunity.
Isn't that just wild? We're talking about a technology that literally goes in and rewrites the genetic code to eliminate a persistent threat. It's like finding a universal undo button for viral infections or giving our cells their own internal personalized security detail.
If we connect this to the bigger picture, it signifies a paradigm shift in how we approach infectious diseases altogether.
Yeah.
Instead of just treating symptoms or blocking replication, we're addressing the fundamental genetic presence of the virus itself, its very blueprint.
Yeah.
This isn't just about managing a chronic illness, it's about learning to program and reprogram the very software of life, offering an entirely new horizon of hope for countless genetic challenges, not just infectious ones.
The idea that we could move beyond lifelong daily drug regimens to a one-and-done treatment that eliminates the need for daily cart, that's the dream scenario, isn't it?
It really is.
It sounds so incredibly elegant and precise, going after that hidden viral reservoir.
It truly is the dream scenario that scientists have been working towards for decades, and the implications extend far beyond HIV. Imagine applying this kind of precision gene editing to fight other persistent viral infections that also integrate into the host genome, like certain herpes viruses, such as cytomegalovirus or Epstein-Barr virus,
Right.
or even hepatitis B, which affects hundreds of millions globally. This technology opens doors to potentially correcting genetic defects that cause diseases like sickle cell anemia, cystic fibrosis, or Huntington's disease, moving us into the realm of truly curative therapies for conditions we once thought were incurable.
So you're telling me we could theoretically train ourselves to be tiny, microscopic superheroes ready to zap any genetic bad guys, like a personalized internal security detail forever on guard.
Tuckle slightly, something like that. The potential is immense, almost breathtaking, but so are the complexities that still stand in our way.
That's a good segue because as exciting as all this sounds, this isn't just a flip of a switch, right? What are the main roadblocks, the significant challenges to making CRISPR-Cas a widespread, definitive cure for HIV? This isn't just theory, it has to work in the human body.
You're absolutely right. While the promise is huge, navigating the road to a clinical cure involves several major roadblocks that scientists are actively working to overcome. The first, and one we've touched on, is viral escape, the virus's inherent ability to adapt and mutate to evade our CRISPR attack.
So the virus strikes back even against these super precise molecular scissors. How does it manage that?
Unfortunately, yes. When using single CRISPR-Cas approaches, meaning targeting the virus with just one cRNA, there's often a high likelihood of viral escape. Remember the cell's NHEJ repair mechanism, that error-prone one we talked about?
Yeah, the messy one.
It's a double-edged sword. While it can be useful to induce inactivating mutations, the virus can also exploit it. If NHEJ repairs the cut in a way that introduces specific small mutations, or INDLs, around the cleavage site, the CAN enzyme might no longer recognize its target sequence for future cuts.
Ah, it changes the target site.
Exactly. It's like the virus quickly changes its specific identifier or the secret handshake it uses, making the same guide RNA useless.
So how do we counter that genetic evasiveness? How do we hit it in a way it can't recover from?
The most promising solution is using multiplex approaches. Instead of a single cRNA, we target HIV with 2 or more cRNAs against a single gene, or even multiple different genes simultaneously.
Hit it multiple ways.
Exactly. Imagine trying to change your disguise when someone's firing paintballs at you from 3 different directions statistically. You're going to get hit and identified. These multiplex approaches have shown much more durable suppression and a significantly lower likelihood of viral escape.
Makes sense.
It's much harder for the virus to mutate and escape when it's being hit in several critical places at once, especially if those mutations would compromise its very ability to replicate. We can also look at combined approaches, targeting both viral genes and host genes, or even adding genes for neutralizing antibodies, as we discussed with the BNAbs, creating a multi-layered defense. And critically, selecting the right Cas system itself matters. Cas-Col-A, for example, is emerging as a particularly strong contender here.
Cas12a.
It's often less prone to escape than Cas9 because its cleavage site is located far downstream of the PAM sequence.
Oh, interesting.
This means that even if the virus mutates near the PAM, the actual cut site is distant, making it more likely that the cut will still happen. This allows for multiple rounds of editing and can introduce more gross, devastating mutations that genuinely prevent viral escape. It really chops up the viral DNA beyond recognition, making it truly non-functional.
That makes perfect sense. Hit it hard, hit it everywhere, and pick the right tools for the job. What's the next big hurdle on this path to a cure?
The next major concern, and one that absolutely demands meticulous attention, is off-target effects. While Cas nucleases are incredibly precise, they aren't perfect. There's a risk they could cut at unintended sites within the vastness of the host genome.
And what are the potential consequences of that?
Yeah.
Unintended cuts in our own DNA sound incredibly serious.
They can be profoundly serious. The risks include unwanted large chromosomal deletions, which could lead to gene silencing, essentially turning off essential human genes.
Yikes.
Even more concerning, an off-target cut could remove or inactivate a tumor suppressor gene, which are genes that normally prevent uncontrolled cell growth in cancer.
Oh, wow.
Or it could even activate a proto-oncogene, which are genes that, when activated inappropriately, can trigger oncogenesis or cancer. This is why incredibly intense safety screens for anti-HIV CRISPR approaches are absolutely warranted, involving extensive genomic sequencing and functional assays to detect any unintended changes before moving into widespread clinical use. We have to be absolutely certain we're not treating one disease for another.
Yeah. So how do we minimize those risks? Are there ways to make CRISPR even more specific, like a laser-guided missile for DNA?
Yes, absolutely. Scientists are actively working on predicting and limiting off-target effects through various sophisticated strategies. This includes modifying the cRNAs, those guide sequences, to make them even more discriminatory, or engineering the Cas proteins themselves to boost their specificity and reduce their propensity for promiscuous cutting.
Tweaking the scissors.
Exactly. The goal is to ensure they only go where they're explicitly instructed with virtually no collateral damage. This involves tweaking the molecular structure of the Cas enzyme to demand an even more perfect match with its target.
Okay, so we've got the tools, and we're refining their precision. But then there's the monumental task of actually getting these molecular scissors into the cells that need them, especially those hidden HIV reservoirs. That's the delivery method challenge, right? It sounds like trying to deliver a tiny package to a needle in a haystack, and that haystack is a human body.
Precisely. This is arguably the single biggest obstacle to a functional HIV cure. Reaching and clearing that multifaceted HIV reservoir. Remember, these are cells, primarily CD4 plus T cells, but also macrophages and dendritic cells that are infected with replication-competent viruses and can persist for extended times, sometimes for decades, even under CART.
Decades. Wow.
They can hide in various tissues throughout the body, lymph nodes, the gut, the brain, the bone marrow, making efficient targeting incredibly difficult. The heterogeneous nature of this reservoir, meaning it's made up of different types of cells hiding in diverse anatomical locations, presents an immense challenge. Clearing all integrated HIV DNA from all infected cells and tissues is an immense undertaking, if not impossible with current methods.
So the virus is not just hiding, it's hiding in a bunch of different places and disguises, like a distributed network of viral sleeper agents. That sounds like a nightmare for delivery. What are the current leading options for getting CRISPR components into these cells?
For viral delivery vectors, adeno-associated viruses, or AAVs, are currently the leading in vivo delivery system.
AAVs.
Okay.
They're popular because of their low immunogenicity, meaning they don't provoke a strong immune response in humans' high target cell specificity, and they typically deliver their genetic cargo as an episome, a separate DNA molecule with limited integration into the host cell's genome.
Sounds good so far.
However, AAVs have a significant drawback. Their packaging capacity is quite limited, to about 4.7 kilobases.
Oh, not much space.
Not much at all. The most frequently used Cas9, SPCAS9, is already about 4 kilobases, leaving very little room for all the necessary regulatory elements, guide RNAs, and reporter genes. Strategies like splitting SPCAS9 into 2 fragments that are introduced via different AAV vectors, then recombining inside the cell have been proposed, but these intricate approaches usually come with significantly reduced editing efficiency.
Right.
It's like having to send a really big book in 2 tiny envelopes.
So AAVs are precise, but they're like tiny carry-on bags. What about other viral vectors that can carry more? Then we have lentiviral vectors, or LVs. These are incredibly versatile. They are able to infect both dividing and non-dividing cells and have a much higher cloning capacity, around 8.5 kilobases.
Okay, that's better.
Which makes them suitable for delivering all CRISPR-Cas components with a single viral vector. LVs are widely used for clinical gene therapy applications, especially when long-lasting transgene expression is required because they integrate into the host genome.
They integrate.
Yes. Crucially, unlike retroviral vectors from earlier, more problematic gene therapies, LVs haven't caused leukemia in gene therapy trials due as much to integration-related oncogenesis effects. However, their prolonged expression of the Cas protein, because they integrate into the host genome, may increase the long-term risk of off-target effects. So additional safety features and careful monitoring need to be explored to mitigate that.
So LVs are more robust for delivery, capable of carrying more, but we need to make sure they're not causing problems by sticking around too long. Are there ways to get the CRISPR components in and out quickly, like a quick hit-and-run mission?
Yes, there are. Scientists are developing what are called non-integrating LV systems that are designed for transient or temporary delivery.
Transient, okay.
These are modified so they don't integrate into the host genome, often by mutating the viral integrase gene or the LTRs, those long terminal repeat regions. This means the CRISPR components are delivered, do their job, and then their presence in the cell diminishes over time, ideally reducing the risk of prolonged KX expression and associated off-target effects. It's a very attractive strategy for safety.
And what about completely non-viral delivery methods? I'm imagining something like tiny molecular envelopes or microscopic drones delivering the goods, bypassing the virus entirely?
You're absolutely on the right track. Non-viral methods are a huge and rapidly advancing area of research. These include lipid-based ribonucleoproteins, or RNPs, where the case protein and its guide RNA are preformed and packaged together in a lipid nanoparticle.
Lipid nanoparticles, like the COVID vaccines.
Exactly like that technology, yeah. Then there are polymer-based RNPs, and even approaches using inorganic materials. These non-viral methods offer a key advantage: transient delivery. This means the CRISPR machinery is present for a shorter controlled time, which can avoid some of the issues associated with viral vectors, such as integration risks and prolonged expression, and can also reduce immunogenicity because you're delivering the functional protein directly, not instructing the cell to make a foreign protein for a long time.
Right.
They're like ready to go, self-contained little molecular bombs.
All right, last major challenge, and this sounds really important for in vivo applications, meaning inside a living human. Immunogenicity. If we're putting bacterial proteins like Cas9 into humans, what our immune systems freak out? They're designed to fight off bacteria after all.
That's a critical point for clinical translation and a very real obstacle. Since the CRISPR-Cas system originates from bacteria, it's highly likely that the human immune system will indeed react against it when it's administered in vivo.
Yeah, it's likely.
In fact, reports already demonstrate that many people possess pre-existing immunity against certain Cas proteins, likely from prior exposure to those bacteria in their lives. This pre-existing immunity can actually frustrate the delivery and effectiveness of the therapy, as our bodies will quickly try to neutralize the foreign Cas proteins before they can do their job. Wow. It's quite ironic our own defenses could be the ultimate roadblock, seeing this amazing cure as a foreign invader.
So our own immune system could be the ultimate roadblock, seeing this amazing cure as a foreign invader. That's quite the biological paradox. How do we get around that?
There are several potential solutions being intensely researched. One is to try and select for non-immunogenic Cas systems, constantly searching for or engineering Cas proteins from diverse bacterial species that our human immune system is less likely to react against.
Find stealthier scissors.
Kind of, yeah. Another approach, though, with its own risks, is the parallel use of immune suppressive drugs alongside the CRISPR-Cas-based therapy to temporarily dampen the immune response while the gene editing takes place.
But those come with their own set of risks, right? Like making a patient more vulnerable to other infections.
Absolutely. It's a careful balance, and we want to minimize those risks. Other strategies aim to shield the case proteins from immune detection. For example, mRNA delivery of Cas instead of directly delivering the Cas protein as RNPs can partly mitigate antibody responses.
Like the vaccines again?
Exactly. Here, you're giving the cell the instructions to make the Cas protein rather than introducing the preformed bacterial protein itself, which can be less immediately inflammatory for the immune system. Or encapsulating RNPs into lipids or polymers can literally shield the immunogenic protein from antibodies in the bloodstream, allowing it to reach the target cells unhindered.
Like a stealth coating.
Pretty much.
Uh.
Overall, it's a multifaceted challenge, but active research is addressing each of these points with clever, innovative solutions. Rational and systematic designs of multiplex approaches, leveraging our knowledge of both host restriction pathways and viral genes, will be essential for achieving long-term suppression and eventually eradication.
So what does this all mean? We've seen how this incredible bacterial defense system, CRISPR-Cas, offers a truly revolutionary way to attack HIV at its genetic core, moving us beyond just managing the disease to actually seeking a true lasting cure. It's about designing intelligent, systematic approaches.
Indeed. It's about designing rational, systematic multiplex approaches, leveraging our understanding of both viral genes and host restriction pathways to achieve long-term suppression and, ideally, eradication of HIV infections. The science is moving incredibly fast, and what we once considered science fiction is rapidly becoming a clinical reality.
Yeah.
We're looking at long-term in vitro and in vivo infection experiments, testing different Cas systems, multiple cRNAs, and various delivery methods to evaluate both efficiency and safety. Our understanding of HIV infection biology is constantly evolving, which in turn helps us refine these CRISPR-Cas strategies, making them more efficient and safer.
And it's not just about HIV anymore, is it? We're talking about a dual strategy. Directly targeting those stubborn viral genes, the ones integrated into our cells, and simultaneously boosting our own body's natural defenses, turning our cells into tiny, superpowered antiviral agents.
Right.
And while there are big challenges viral escape, those tricky off-target effects, figuring out how to deliver it precisely to every single hidden reservoir cell, and dealing with our own immune system, the fact that these are all actively being researched and addressed means we're in a completely different ballpark now. The ingenuity of the solutions is truly inspiring.
This is truly a dynamic field where every new discovery refines our ability to precisely manipulate life's most fundamental code.
This isn't just a science story, it's a human story. What other diseases, once thought incurable, might be next on the CRISPR chopping block? What future breakthroughs, what astonishing leaps in human health might emerge from this deep understanding of life's fundamental code? It makes you wonder how much more our bodies are capable of, if only we learn to speak their language.
This raises an important question. As we gain the unprecedented ability to precisely edit genomes, to truly rewrite the software of life, how will we choose to wield this immense power, not just for cures, but for shaping the very future of human health and our interaction with the microscopic world around us?
It's a future where the tiny, elegant mechanisms of bacteria could unlock cures for humanity's biggest challenges. What an incredible deep dive this has been.
Truly remarkable. There's always more to learn, more questions to ask.
Until next time, keep digging, keep questioning, and stay curious.
Because the answers are out there.
With a playful sigh. Benny, any last puns for the road?
Oh, I've got one. I guess for HIV, we're really getting to the genius is the problem, huh? Cutting to the chase.
Groans playfully. I knew it. That's our deep dive for today. See you next time.